predisposition to developing pancreatitis. Note in the upper panel that the common bile duct and the major pancreatic duct enter the duodenum together. And as you remember, gallstones can traverse the biliary tract through the common bile duct and cause biliary pancreatitis. They probably do so because they cause obstruction at this common orifice of the common bile duct and the pancreatic duct. The major cell types in the exocrine pancreas are shown here. The Astner cells can be easily identified by their prominent zymogen granules. And the duct cells are more difficult to see even at, on this micrograph. These are small, flat, cuboidal cells. The blue cells are called centroastner cells. I put them there for completeness. They're at the junction of the Astner cells and duct cells, and they may have roles in sensing secretion, coordinating secretion, and as progenitor cells in the pancreas. The ultrastructural morphology is better shown here in these electron micrographs. Shown to your left is an Astner cell as compared to a duct cell, and even for non-scientists, it's pretty easy to see that these are quite different. The large dark granules are the sites in which we store our digestive enzymes, and they're going to be secreted into the lumen when we, shown above, at the time of a meal. At the base of the Astner cell, you can see this prominent lacy web surrounding the nucleus, and that's the endoplasmic reticulum. That's where protein synthesis begins. The Astner cell, exocrine pancreas, has probably the highest average rates of protein synthesis in the body, producing digestive enzymes. You can see the difference easily between the Astner cell and duct cell to the right. There are some dark structures. These are not as dark as zymogen granules. They tend to be linear, and these are mitochondria. And mitochondria in an epithelial cell mark them as cells that are likely to be involved in ion secretion. Ion secretion, such as the chloride and bicarbonate made by the exocrine pancreas, are very expensive in terms of ATP production. And as a reflection of that, cells that are producing lots of iron, such as parietal cells in the stomach, will be dramatically enriched with mitochondria compared to non-ion secreting cells. I compared that's interesting. These are backwards. I can't believe them. Okay, be sure to change those in your note. It's funny when I even looked at this morning. Okay, well, the exocrine is actually on your left and the endocrine on your right. And Melina Bellin will certainly know the difference in, between those two. So anyway, the Astner cells are responsible, as mentioned, for generating proenzymes and enzymes, where the duct cells produce water and electrolytes and bicarbonate. The islets, shown to the right, this well-circumscribed area of distinct, rather monotonous-looking cells, generate insulin, glucagon, and somatostatin. The pancreas represents one of the three portal systems in the body, the other two being involved with the liver and the brain and pituitary. What that means is, as shown here, high concentrations of islet hormones, the islet to your right, reach the exocrine pancreas. So about half of the pancreatic Astner cells are bathed by this secretion from the islets that have very, very high concentration of islet hormones. We're not certain of the importance of this. It certainly, I think, has a homeostatic and regulatory role. One possible reflection of it is the recent description that patients that have dysfunction of their islets, particularly when they develop earlier in life, develop extensive fibrosis of the pancreas, some evidence that they can have decreased exocrine secretory function. So both the endocrine and exocrine pancreas respond to the stimulation into a meal. The exocrine, which again is mislabeled, is shown on your left, responds to sight, smell, and taste of food, food in the stomach, duodenal pH, and nutrient content, whereas the primary stimulant for the endocrine pancreas to your right is going to be blood glucose levels. Eating stimulates secretion into the duodenum, and you can see there's, as shown to the left, during the meals there is some secretion of pancreatic enzymes into the small intestine. You can see it has a phasic nature, but there's a dramatic increase when one eats a meal that starts even when one visualizes the food, smells it, and tastes it. Secretion stops. If you look to the far right, you'll see that there's a termination of secretion as measured by trypsin output into the small intestine. And that's actually an active process. So there are signals that come from the distal small intestine that tell the exocrine pancreas this is the time to stop secreting. You don't want to waste pancreatic secretion if there's no food there. So the off button is located at a site that's quite distal to the pancreas. We'll talk about what turns things on in the gut momentarily. So one of the things that I found always interesting and fascinating is the degree to which the pancreas is stimulated before food even meets the intestine. So we divide this into phases: cephalic, gastric, and intestinal phase. The stimulants are shown there. So even the site of food can stimulate pancreatic secretion over 20% of its maximum value. Stimulation when food is in the stomach, and of course the maximum stimulation when it's in the small intestine. So I think this has teleologic importance, which reflects the fact that until very recently, we were in a nutrient, a low nutrient, condition, and we really developed physiologic responses that would maximize nutrient utilization. One of the ways we do that is by stimulating the secretion of the pancreatic enzymes before food gets there. They can be ready to digest food as soon as it arrives. There are very specific pathways for stimulating secretion from the pancreas, and they vary between stimulation of pancreatic enzyme secretion versus bicarbonate and fluid secretion. There's specific receptor sensor cells, neuroendocrine cells in the gut, in the proximal gut, and the duodenum that sense the nutrients. The major stimulations for the secretion of the enzymes are free fatty acids and specific peptides and amino acids, and they stimulate a population called I cells, and these work primarily through a neural pathway to stimulate the pancreas, possibly some local direct stimulation. Acid and free fatty acids stimulate another type of neuroendocrine cell in the duodenum called S cells. These releases a hormone called secretion that also stimulates pancreatic secretion. Bicarbonate secretion is important for neutralizing acid in the small intestine. As shown in the left, as we increase the flow rate or the stimulation of the pancreas, the concentration of bicarbonate increases rather dramatically, and chloride levels fall in what's being secreted into the duodenum. One of the interesting features of this response is that it does have a threshold, and this is somewhat unusual for physiologic responses in humans. And there seems to be a fairly tight setting, as shown in the panel on the right. When duodenal pH hits 4.5, that is what's required to turn on the secretion of bicarbonate into the intestine. So another interesting feature of the pancreas, both endocrine and exocrine, is the enormous reserve, and shown here is data on the exocrine pancreas. So normally we excrete about 6 to 7% of the fat that we intake from our daily diet. And that's shown by the pink marker it measures lipase output and fat excretion, and that's marked the normal range. Now, if you decrease lipase output, as shown on the x-axis, you can see one has to go below 90% of lipase output before fat excretion starts going up. What that means is when we see a patient with pancreatic exocrine insufficiency, they've had a tremendous amount of loss of pancreatic reserves. I think it may be spoken to at the discussions today, but one of the observations made by clinicians over the past couple of years is the development of pancreatic insufficiency after acute pancreatitis, and exocrine and endocrine insufficiency is actually much more common than we had thought in the past, and it means that there's a large portion of the functional reserve of the gland is being lost. The content of the digestive enzymes are shown here. As shown to your left, many, and including all proteases, are made in an inactive form called zymogens or proenzymes. Some are made, shown to your right, as active enzymes. The proteases are normally just activated after reaching the small intestine. They first are, this activation process is first initiated by the clipping by a protease in the intestine called enterokinase of trypsinogen, which converts it to trypsin. And as shown here, trypsin can then, by limited proteolysis, catalyze the activation of all the other zymogens secreted into the small intestine. Finally, we think one of the mishaps that occurs to initiate acute pancreatitis, which will be a topic of discussion in subsequent session, is the activation of these digestive enzymes actually within the pancreatic acinar cell. So we've gone a long way to protect against that. As I mentioned, most of the digestive enzymes, all proteases, are secreted as inactive zymogens. They're packaged, trypsinogen is packaged with an inhibitor in the zymogen granule. There's mechanisms for degrading any active trypsin that's generated in the acinar cell. They're segregated in these membrane-bound compartments, so hopefully they don't escape if they're activated. And finally, enterokinase is restricted to the small intestine. I think I'll stop at that point, and thank you all for your attention.